On the last page of your fill in the blank study guide, you'll notice that on about number 40, it's going to start covering some information about bacteria and viruses that we haven't covered. You can cross out 40. That will definitely not be on the exam. And if for some reason I forget to omit it from the exam, I will go ahead and grade the exams and then change the percentage so that that will not figure in, okay? Also, number 42 you can cross out. That will definitely not be there. Okay, let's look at number 41 first. It says the chromosomes of bacteria. If you are familiar with the structure of a bacterium, it has a cell membrane and it has a cell wall. Do you remember which group of bacteria have cell walls made of peptidoglycan? Which ones has, oh, we did that in biology 1, not biology 2. Okay, so cell membrane is on the inside. Cell wall is on the outside. And inside of the cell, you will find a single circular piece of DNA called a plasmid, okay? Now, recall that bacteria are prokaryotes. What is a prokaryote? Do they have nuclei? Prokaryotes do not have nuclei, okay? So this bacterium does not have a nucleus. Prokaryotes do not have nuclei. And they do not have membrane-bound organelles.
Which one is this on?
This is on the second study guide, the one with the fill in the blank. It was actually multiple choice and there was blanks on the left-hand side for you to fill in. Did you get this?
This one?
Yes.
What number is it?
Well, we're looking at various numbers right now. I'm just going to tell you a little bit about bacteria and viruses and then we'll look at the particular numbers. Okay? So, this does not have membrane-bound organelles such as chloroplasts, mitochondria, but it does have some organelles. It has ribosomes. Thank goodness, if it didn't have ribosomes, it wouldn't be able to have proteins. The cell would not be able to make proteins. So there are ribosomes in bacteria. And bacteria most often produce asexually, but they have a special mechanism for exchanging genetic material, which could be considered sexual because they're exchanging genetic information, but they're not going to produce a new cell by this means. So let's say these are two little bacteria hanging out together, and they pass a little tube between them, and through that tube, this one's going to pass some of its DNA, and this one will pass some of its DNA over. So there is a DNA exchange. And that DNA exchange in bacteria is called conjugation. Okay? So conjugation is a DNA exchange, but bacteria also reproduce asexually by fission. Basically, all that happens during fission is the DNA will replicate and then the bacteria will split in half. So you'll have two bacteria where you once had one. Okay? Now, there is a big invader of bacteria and of all living organisms, Well, you know that bacteria invade us, but also we can get invaded by viruses, and that's why I wasn't here last time, is because I was not feeling very good because I was sick. And bacteria sometimes get invaded by viruses as well. So this bacterium has an invader on it from outer space. No, it's not from outer space, but it looks like it might be. It's a virus, and this particular virus is called a bacteriophage because it invades bacteria. So bacteria obviously has to do with bacteria. The phage means basically to eat. Bacteriophages are a kind of virus that attacks bacteria. A kind that attacks bacteria. Okay? So basically all a virus is made up of is the outside portion, I believe, is called a capsid. And a capsid is basically a protein coat. And the only other thing that it's made up of is genetic material, nucleic acids, for example, DNA. So inside the virus is the DNA. What are the two parts of a virus? The protein coat and the DNA. Okay? Or it could be RNA. Does that remind you of Jimmy Neutron? No? Okay, so this is the bacterium that it's going to invade. What do I mean by invade? Well, what happens is this DNA gets inserted into the bacterium. So that DNA comes down. And remember, we said that the bacterium has a circular piece of DNA in it. So here's its circular DNA. Viruses can do one of two different things. One, they can actually take their DNA and insert it into the bacterium's DNA. So right here, this darkened portion, right here, is the viral DNA. Then guess what will happen if this bacterium replicates? What will happen if the bacterium replicates? What's going to be part of its new DNA in each of the cells that it makes? That bacterium is actually going to make more viral DNA for the new cells that it's producing. So see how a virus can take over your body? Then what happens? Well, if this is this particular type of virus, which is called a lysogenic virus, lysogenic viruses have a cycle that's like this. They invade host cell. That's step one. Step two is viral DNA becomes part of host DNA. The host replicates, and then the cells can lyse. So what do I mean by lyse? It just means that they're going to break open. So basically, once cells are reproduced, these cells break open, okay? And that viral DNA goes free and it goes and invades in another place. Okay, that's my understanding of it. The other type of virus, basically, it doesn't follow all these steps. It doesn't even get to this stage where the host cell will replicate. It'll just instantly break out. It'll destroy the host cell, and then it'll go and destroy another host cell. That's a lytic virus. So that's pretty much everything. There's one last thing I want to mention about bacteria, and that is that there's a very important type of bacteria to human beings. It's a beneficial bacteria that lives in the roots of plants. So you might pull out a weed one day and notice that on that weed's roots there's like a little knot, or there's several knots. And these little knots or nodules actually are filled with what are called nitrogen fixing bacteria. The nitrogen fixing bacteria take atmospheric nitrogen, which is in the air, and they fix it into a form plants can use. And this is awesome because if you're a farmer, for example, and you're planting a crop of corn that takes a lot of nutrients out of the soil, you have more options than just applying an artificial fertilizer. You don't have to go to the store and buy an artificial fertilizer. Another option would be for you to plant another crop for a while that has these nitrogen fixing bacteria in the roots, and that's going to actually enrich the soil because nitrogen is the main component of a fertilizer. So you can plant a legume, maybe alfalfa or clover or some other plant that is going to have these things in the roots, helping the plant to grow and also at the same time enriching the soil. Okay? Are there any questions so far? So let's look back at your study guide questions and see if we can answer these questions based on what we've learned. Number 41. The chromosomes of bacteria. What are they? A, do they contain numerous types of organelles? The only organelle we spoke of was ribosomes. So I'm going to just X that answer out. I don't think that's it. Are the chromosomes divided into compartments? No. Do they vary in number depending on the species of bacteria? They might, but we said that usually, if not always, they contain only one single circular piece of DNA called a what? Class? Look back to your diagram. What is that single circular? Plasmid. That's a plasmid. Okay. So number 41 is D. 43. Cell organelles that E. coli and other bacteria have in common with eukaryotes. We said they don't have, no bacteria, no prokaryotes have chloroplast or mitochondria because those are membrane-bound organelles, nor do they have a nucleus. So the answer is D, ribosomes. Bacteria cells have what? Do they have a cell wall only? No. Do they have a cell membrane only? Do they have both a cell membrane and an outer cell wall? Yes. Is the cell wall inside their cell membrane? No, it's outside. So the answer for that one is C. Oh, here's one we didn't cover in the review. Cyanobacteria are photoautotrophs because they require, I don't like this question because they're going to require light and they're also going to require carbon dioxide. So I think there's two answers on that one. Miss Weedy might tell me that she wanted one of those answers for a specific reason. Maybe it's because it's photoautotrophs versus I think there's a chemical autotroph. I can't remember if they're called chemoautotrophs or not. So maybe that was the specification. I'm going to toss that one out. The nitrogen-fixing bacteria do what? Do they repair nitrogen-damaged soybean roots? Or do they damage the environment by using atmospheric oxygen to produce toxic nitrogen? No. Do they convert atmospheric nitrogen to a usable form of nitrogen? Okay. Do they convert ammonia in the soil into nitrogen gas? No. C is your best answer. It's the only answer.
For 46?
For 46, yes. 47, bacterial cells such as E. coli transfer pieces of genetic material in a process called, conjugation. Conjugation. C. Oh my gosh, sometimes life is so hard and I just want to close myself in an endospore and not have to respond to anything in the world. That's how bacteria feel sometimes. They just close themselves off. They don't have to worry about food or the weather or anything, and they can wait years like that. And then when the conditions improve, they burst out of their protective endospore and they start reproducing. So, an endospore is all about what? D, allow certain species to survive harsh environmental conditions. Very good. A typical virus consists of, is it A, protein coat and cytoplasm core? No. Is it carbohydrate coat and a nucleic acid core? No, it's not a carbohydrate coat. It is a protein coat and a nucleic acid core. Remember, the two examples of nucleic acids are DNA and RNA. The answer is C. Let's check and see what D is. Polysaccharide, that would be a sugar coat, and a nucleic acid core. That's not correct. Look at the diagram. This is the picture I drew for you earlier, sort of. What is letter A?
Protein.
A protein coat. Okay, what is C?
DNA.
DNA. Okay, so for number 50, they're asking which part represents the protein. That is the letter A. Don't get confused and put letter A here, because that says structure B. It's letter B, structure A. Don't know why anybody would want to do that to us and try to confuse us. The function of a bacteriophage's tail and tail fibers is to inject what? Are they going to inject the protein? No. What would be the purpose of that? The cell cannot reproduce with protein. Is it going to insert enzymes? No. Glycoproteins? No. Viral DNA? Yes, it's D. The capsid of the virus is the protective outer coat. I think that is the answer, A. And you can see that something was skipped here, 53, 54. I think I already deleted those from here. And the last question on your study guide. The cycle of a viral infection, replication, and cell destruction is called the viruses. Remember I told you there's two types of viruses, lytic and lysogenic. So I think the one that has the viral infection, replication, and cell destruction is called the virus's lysogenic cycle. Okay? Are there any questions? Any other questions from the study guide in general that you couldn't answer on your own?